Hello, good afternoon, students. Today we are going to discuss on cell division. Last classes we have dealt with the ultrastructure of the animal cell. So we have seen what is cell, what are the different parts of a cell, how the cell is made up of, and the different organelles that are present in the cell. Today we are going to talk about the cell division as well as the cell cycle. Now, before we start with this, we should know actually what do you mean by cell division? Why is it necessary for the cell to divide? And what do you actually you mean by a cell cycle? Now, cell division actually is nothing but it is a process in which the cell, which is actually basically we call it as a parent cell, what it will do, it will try to divide to form two new cells, which is referred to as the daughter cells. So here in the cell division, actually we can define cell division as nothing but a process in which the parent cell will divide into two cells, which are called as nothing but the daughter cells. Now coming to cell cycle, what do you mean by cell cycle? Cell cycle actually is nothing but it is a repeating series of events that include the growth, DNA synthesis, as well as the cell division. So that is what we call it as a cell cycle. So cell cycle just does not include not only include cell division, but it also includes the growth. It also includes the synthesis of DNA as well as a cell division. So these two actually go simultaneously. So both cell division as well as the cell cycle play a very important role. So in the before we actually proceed to the cell division, now first we should know actually about the cell cycle. What are the different phases of the cell cycle? What actually happens in the cell cycle? Now the cell cycle actually basically was discovered by the scientists called as Prevost and Dumas in the year 1824, while in the 1824 while studying the cleavage of the zygote of frog. So when they were studying the cleavage pattern in the frog zygote, then they frog zygote actually they found out the cell cycle, and this was actually given by the Prevost and the Dumas. So they said actually they described the cell cycle as nothing but it is a series of stages a cell will pass through to divide and also to produce the new cells. That is what actually is called as the cell cycle. So we all know, we know now that the cell division is what? Cell division is nothing but it is a process in which the parent cell will divide into two cells, which we call it as a daughter cells. Whereas cell cycle actually is a repeating process, which includes the growth, the DNA synthesis, as well as the cell division. The cell cycle, first of all, was discovered by Prevost and Dumas in the year 1824 when they were trying to actually study the cleavage pattern in the frog zygote. And they describe the cell cycle as nothing but a series of stages a cell passes through to divide and also produce a new cells. So thus the entire process with the help of one single parent cell, a new cell population grows and develops is known as nothing but a cell cycle. So the entire process we call it as nothing but a cell cycle. Now we'll see what are the different phases of a cell cycle. Now the different phases of a cell cycle are nothing but it refers to the series of events, just as I told you, that takes place in a cell leading to the maturity as well as division. So these events actually include duplication of its genome, synthesis of the cell organelles, followed by the division of the cytoplasm. So a typical eukaryotic cell, if you take, okay, the cell cycle in the typical eukaryotic cell is divided into two main phases. One is called as the interphase and the other one we call it as nothing but a mitotic phase. Now, which is also called as the M phase. So the different phases of the cell cycle are divided into two. One is called as the interphase and the other one you call it as a mitotic phase. This is also called as the M phase. Now we'll see one by one what also. See, this is how actually the what do you say the cell cycle looks like. This is the interphase. Interphase actually we call it as nothing but a resting phase, but actually speaking it is not a resting phase. Here it is in this particular phase only the cell actually will prepare itself for the division. It will not rest, but at the same time what it will try to do, it will try to synthesize whatever is required for the cell division. So it will prepare itself for the division. So that is what we call. Now this interphase is followed by all these things. So S stands for the synthetic phase, G2 is the growth phase 2, M is the mitotic phase, and G1 actually is the growth phase 1, and GO actually is referred to as nothing but a resting phase. So that is how actually it is divided into the different phases. Now we'll see one by one the interphase. First one. So basically, typically we can say a eukaryotic cell cycle is divided into two phases. One is called interphase, and the other one you call it as nothing but a mitotic phase. Now we'll see one by one. What do you mean by interphase? Now, interphase, also known as the resting phase of the cell cycle. We can also call interphase as the resting phase of the cell cycle. Now, it is the time during which the cell will prepare itself for division. Just now I have told you, no. So the cell will not rest. No doubt it is called as a resting phase, but it will not rest. It is during this particular period only the cell will prepare itself for division by undergoing both the cell growth as well as a DNA replication. So it occupies around 95% of time of the overall cycle. So about 95% of time of the overall cycle. So it will be actually going on, preparing itself for the cell division, but it will not rest. And this interphase is further divided into three phases. It is called as a G1 phase, that is the growth phase you can call, or the gap 1 phase also you call. Then you have S phase, which is called as the synthesis phase or the synthetic phase. And you have got the G2 phase, which is called as a gap 2 or the growth 2 phase. So this is how. So basically, the cell cycle is divided into two phases. Just now I have told you, one is called as interphase and the other one is called as the mitosis. Now, in the interphase, though it is called as a resting phase of the cell cycle, but it is this particular period where the cell will try to prepare itself for division by undergoing both the cell growth as well as the DNA replication. This interphase is divided into three phases. One is called as G1 phase, second one is the S phase, then the third one is called as a G2 phase. Now, coming to the G1 phase, G1 phase actually is a phase of the cell division between the mitosis as well as the initiation of replication of the genetic material of the cell. So in this, during this particular period, it is a phase actually which occurs between the mitosis as well as the initiation of the replication of genetic material, where DNA replication will take place, where DNA try to actually divide to produce new DNA material. So it is a phase between the mitosis as well as the initiation of the replication of genetic material. So during this phase, the cell actually is metabolically very active and it continues to grow without replicating its DNA. Only initiation will take place, but it will not replicate. So that is why it is called as the G1 phase. So it is a phase between the mitosis as well as the initiation of the replication of the genetic material. So here, during this particular phase, the cell is very, very active and it will continue to grow without the DNA, without the replication of DNA. Now, the second phase is called as nothing but the S phase, which is also called as the synthesis phase or the synthetic phase. Now, during this particular period, DNA replication will take place. And if the initial quantity of DNA in the cell is denoted as 2n, then after replication, it becomes 4n. That means it will become double. However, the number of chromosome does not vary because if the number of chromosome during G1 phase was 2n, then it will remain 2n at the end of S phase also. So, the centriole also divides into two centriole pairs in the cells, which contains the centriole. So like this, so that is what we call it as S phase is nothing but called as a synthetic phase or the synthesis phase. Here during this particular phase only the replication of the DNA will take place. And here during this particular period, what happens is if the initial quantity of the DNA is denoted as 2n, then after replication, actually it will become 4n, no doubt. But the number of chromosomes will not vary. It will not vary. It will remain as it is. Okay. Apart from this, the centriole also will divide into two centriole pairs in the cells which contain the centriole. Now, next phase is called as the G2 phase, which is also called as the growth phase 2 or the gap phase 2. Now, during this particular phase, the RNA, proteins, as well as other macromolecules required for the cell multiplication of the cell organelles, spindle formation, and the cell growth are produced as the cell will prepare to grow into the mitotic phase. So this is called as nothing but the G2 phase or the growth phase 2. So during growth phase 1, what happens actually? It is nothing but a phase between mitosis and initiation of replication of genetic material. Only the the genetic material will get initiated for to go for replication, but it will not replicate. It will become active and it will continue to grow. That's all. But in S phase only, the DNA replication will take place. Whereas in the growth phase, that is called as the G2 phase or the gap 2 phase, during this particular phase only, the RNA, proteins, as well as the other macromolecules which are required basically for the multiplication of the cell organelles, spindle formation, as well as for the cell growth, they are produced. Because as the cell is preparing itself for the mitotic phase, it requires all these things. So that is the reason why the RNA, proteins, as well as other micromolecules also are being produced in this particular phase. Okay. Now, coming to some cells like cardiac cells in the adult animals, they do not exhibit division, and some others only will divide to replace those cells which have been either damaged or lost due to the cell death. So, such cells which do not divide further attain an inactive geophase, also known as the quiescent phase. After they exist the G1 phase. So these cells remain metabolically active, but they do not divide unless called upon to do so. So here, what they mean to say here is some cells like the cardiac cell, that is the heart cells in the adult animals, they do not exhibit division. That means they do not undergo the division, and some other cells also will not divide. They will no doubt they divide only to replace those cells which have been either damaged or have been either lost or destroyed. So such cells, what do? They do not divide further, but they attain an inactive phase. That phase we call it as nothing but a geophase or the zero growth phase, or you can also call it as a quiescent phase. Quiescent phase. So here, these cells, though they remain metabolically active, but they do not divide unless they are called upon to do so. So this is what is called as nothing but the geophase. So after the G2 phase, you have a small phase which is called as the you can say as the nil growth phase or the zero growth phase or the quiescent phase. So here what do they do is the cells which do not divide further, they try to actually remain inactive in this particular phase. So such a phase you call it as the geo phase or G0 phase, you can say, or what do you say, the quiescent phase. Okay. Now next is start the mitotic phase. Now mitotic phase is nothing but called as the M phase. Now as I told you, the cell cycle actually involves two phases. One is called as an interphase and the other one is called as a mitotic phase. In the interphase, we have seen it is, though it is called as a resting phase, but the cell will not rest. It will undergo or it will prepare itself for the cell division. So this is again divided into three phases. Just now what we have seen, that is called the growth one phase or gap one phase. Then you have got the synthesis phase or the synthetic phase S phase, and you have got the G2 phase or the gap 2 phase. So in G1 phase, actually only the initiation of replication, DNA replication of the genetic material will occur, but it will not undergo replication. Only the cell will become active and it will continue to grow without replicating its DNA. Whereas in synthetic phase, actual replication of the DNA will take place. And apart from it, the centriole will also divide into two centriole phase in the cells. And in the G2 phase, the RNA, proteins, and other macromolecules actually required for the multiplication of the various cell organelles, spindle formation, they are being produced in this particular phase. Now, after this, some cells actually which do not exhibit division, like heart cells and all those things, what do? Is they do not undergo division, but they actually attain an inactive phase. That phase you call it as a you can call it as a nil growth phase or the zero growth phase, or you can also call it as a quiescent phase. So during this particular period, though the cells remain metabolically active, they remain metabolically active, but they do not divide. So for the purpose of reproduction, they just remain inactive unless until they are called upon. So that is what we call it as nothing but the zero phase. Now next coming to mitotic phase. Now we will see what is the mitotic phase. Now mitotic phase actually is the phase of equational division. Okay, as the cell undergoes a complete reorganization to give birth to a progeny that has the same number of the chromosomes as the parent cell. So you all know actually there are two types of cell divisions if we want to know. That is what we call it as mitosis as well as meiosis. Now mitosis actually occurs in all the somatic cells or the body cells, whereas meiosis occurs in the it is actually a reduction division where the chromosome number is reduced to half, and here it occurs in the germ cells or the sex cells. Now coming to the mitotic phase. Now mitotic phase actually is called as the equational phase because here the cell will undergo a complete reorganization to give birth to progeny which possesses the same number of the chromosomes. So the other organelles also are also divided equally by the process of cytokinesis, which is preceded by mitotic nuclear division. So in this process, what happens is a eukaryotic cell will separate the nuclear DNA and the chromosomes and divides into two different but similar set of nuclei. The chromosomes are pulled apart by a mitotic spindle, which is a specialized structure consisting of the microtubules. So in this mitotic phase, basically what happens is the cell will the eukaryotic cell will separate the nuclear DNA and the chromosomes. It will separate the nuclear DNA as well as the chromosomes and divide into two different but similar set of nuclei. And these chromosomes actually they are pulled apart by means of the mitotic spindle, which is actually a specialized structure consisting of the microtubules. Now this we'll see. So this is what we call it as nothing but the mitotic phase. Now we'll see the how these mitotic phase occurs and what are the different stages of this mitotic phase or the mitotic division. Now, coming to the mitotic phase, they are divided into four stages. They are called as the prophase, metaphase, anaphase, and telophase. So, you find four different types of phases in the mitosis. They are called as the prophase, metaphase, as well as the telophase. Now, coming to the prophase. Now, the prophase actually is the first stage of karyokinesis of mitosis. So, you all know actually whenever cell division takes place, first the nucleus will divide, followed by the division of cytoplasm. So the division of the nucleus, we call it as nothing but karyokinesis, whereas division of cytoplasm, we call it as cytokinesis. So first in all these things, first the nucleus will divide. So when once that is over, then the cytoplasm also will become constricted in the middle, and then it will divide, giving rise to different types of cells. Okay. Now we'll see do that later. Now first we'll see what is prophase. Now prophase, they say actually it is the first stage of karyokinesis of mitosis. Now, actually here it is marked by initiation of condensation of the chromosomal material. So during this particular phase, what happens is the chromosomes which are present in the form of reticular network, as we say, which is present in the nucleus, it will try to get condensed. So condensation of the chromosomal material will take place, and this chromosomal material it will condense to form compact mitotic chromosomes. Because initially before the cell division or cell division, what happens is the chromosomes actually will not have a particular defined shape. It will be in the form of always in the form of a chromatin network or the chromatin reticulum. But only at the time of cell division or whenever the cell wants to divide or is ready to divide, that time only it will try to attain the shape. So that is what they say, that in the prophase stage, what happens is the condensation of the chromosomal material will take place. That is, the chromosomal material will condense to form the compact mitotic chromosomes. So here the chromosomes, they can be observed as composing of two chromatids attached together at the centromere. So if you observe the structure of the chromosome, see here. If you observe the structure of the chromosome here, actually it consists of two chromatids and in the center which are held together by means of the centromere or the kinetochore. So that is how. So it is composed of two chromatids. They say that actually during this particular phase, the chromosome consists of two chromatids attached to a centromere or the kinetochore. Now, the centrosome which had undergone duplication during interphase, what it will do, it will begin to move towards the opposite poles of the cell, and each centrosome will radiate out microtubules called as the asters, and the two asters together with the spindle fibers forms a mitotic apparatus. So that is how it will take. So we all know actually centrosome. Now centrosome actually is present only in the animal cell. It is absent in the plant cell, and this centrosome is located actually just beneath the nucleus. It is located beneath the nucleus. If you see the structure of a cell, you will observe the centrosome location to be underneath the nucleus. So this actually centrosome will undergo duplication only during the interphase stage. So what it will try to do now here in this prophase stage is it will begin to move towards the opposite poles of the cell. So it will begin to move towards the opposite cell, and each centrosome will radiate out the microtubules, which are called as the asters. And these two asters together, they form the spindle fibers. The two asters together with the spindle fibers, they form the nothing but the mitotic apparatus. See, this is one and this is one. So they move actually towards the opposite pole. So this is how actually the centrosome, which has undergone duplication during during the interphase, what it will try to do, it will try to begin towards the two opposite poles. And each centrosome will radiate out these microtubules, which are called as asters, and these two asters, okay, together with these spindle fibers, you call them as nothing but the mitotic apparatus. So these are the changes that occur during prophase. Now coming to the metaphase. Now in metaphase, complete disintegration of the nuclear envelope. So here the nuclear envelope will totally get disintegrated. Hence, the chromosomes are spread throughout the cytoplasm of the cell. So in this stage, what happens is the nuclear membrane will get disintegrated totally, and the chromosomes now will be released. So when once there is no nuclear membrane, means the chromosomes actually will get distributed throughout the cytoplasm of the cell. Then this is the stage at which the morphology of the chromosome is also being studied. So easily you can identify the chromosome structure here, because at this particular stage, the metaphase chromosome is made up of two sister chromatids. It is made up of two sister chromatids which are held together by means of the centromere. So we can say actually during this particular phase, the actual structure of the chromosome can be identified and it can be studied. And according to them, the metaphase chromosome actually they say it consists of two sister chromatids which are held together by means of the centromere. And the spindle fibers are attached to kinetochores of the chromosomes. So what do they do? The spindle fibers which are present towards the two opposite poles, no, they will get attached to the kinetochores of the chromosomes in the chromosomes. And the chromosomes actually they are moved towards the spindle equator and they get aligned along the metaphase plate through the spindle fibers to both the poles. So this is what these changes that occur during the metaphase. Now we'll see the structure. Now see here. This is a metaphasic structure. So these are all the chromosomes. So they are arranged actually in the center, in the equator. And here these are nothing but the two asters. So the spindle fibers that are migrating, they are nothing but the microtubules. So they try to get adhered. So these microtubules actually they are attached to, see here, these microtubules are attached to the centromere of this what do you say, the chromosome. So that is how it will take place. Now in the next phase is called as nothing but the anaphase. Now, the anaphase stage is characterized by these events. One is the centromeres will split and the chromatids will separate. So here what happens, the centromere. What is a centromere? The centromere actually the centromere is nothing but a kinetochore which will help the two sister chromatids. So during this particular stage, what happens? The centromeres will get splitted up. And as a result, what happens? The two chromatids will get separated. So the centromere will split and the two chromatids separate. And the chromatids, what do? They move towards the opposite pole. So that is how from the equator, now they grow towards the opposite poles. So that is why. So one will come towards this side, the other one will go towards the opposite side. So like this actually happens during the interphase, anaphase stage. Now the last one is called as the telophase. Now in telophase, the chromosomes that have reached their respective poles, they decondense and then they lose their individuality. So when once they reach towards the poles in telophase, the chromosomes that have reached towards the two poles, what do? They will actually lose or they decondense. Means actually, if you see in the prophase, what happens? They try to condense and assume a certain shape. But here in this case, in telophase, they try to decondense, okay, and lose their individuality. They don't, give a particular shape again. So they try to lose their individuality and they try to decondense. So here what happens? Again, when once they decondense, the nuclear envelope will develop around the chromosome clusters at each pole, forming two daughter nuclei. And the nucleolus Golgi complex as well as the ER, that is endoplasmic reticulum, again will get formed. So like this, the telophase will take place. So in this particular phase, that is the telophase, the chromosomes actually that have reached their respective poles, what do? They do? They try to decondense and they try to lose their individuality. So as a result, what happens? The nuclear envelope will develop around these chromosome clusters at each pole, forming the daughter nuclei. Apart from this, the nucleolus Golgi complex as well as endoplasmic reticulum also try to get reformed. So these are the four different phases that you see in a what is that mitosis. So what are those? Prophase, metaphase, anaphase, and telophase. So this is how it looks like in the telophase. Now, coming to cytokinesis. Now, just I have told you that actually cytokinesis is nothing but the division of nucleus. So, when once the nucleus divide, now the cytoplasm also should divide because the parent cell has to get divided into two daughter cells. So, how can it get converted into two daughter cells unless and until the cytoplasm divide? So, the cytoplasm also has to divide. It also has to undergo division to form the two daughter cells. So the karyokinesis is always followed by cytokinesis. First, the nucleus will divide, followed by the division of the nucleus, cytoplasm. Now we'll see what happens during this particular cytokinesis. Now the cytokinesis will start just after telophase, which denotes the division of the cytoplasm to form two daughter cells. So what happens is, see, when the two daughter nuclei are being formed and they are towards the two poles, that is in the telophase stage, the cytoplasm actually, which is present in the middle, it tries to constrict. It tries to come closer and it tries to get constricted. So that constriction actually will deepen, and it will try to divide cell into two daughter cells. That means the parent cell will get divided into two daughter cells. So that is how the cytokinesis will take place. So thus it marks the completion of the cell division. So when once the two daughter cells are being formed, then the cell division actually is total. That means the cell division will be completed. So that is how the mitosis will take place. So mitosis is a very, very important what is a cell division that we see in almost all the cells. Basically, it is mostly seen in the what is the somatic cells, somatic cells, that is the body cells, and it is divided into four phases. That is called as nothing but the prophase, metaphase, anaphase, and telophase. Now we have seen what are the different what is say the stages, and in each stage what actually happens. So first the nucleus will divide. When once the nucleus divide, followed by the division of the cytoplasm should be there because it has to be get divided into two daughter cells. Now we'll see what is the significance of mitosis. Now why mitosis is essential? Why there is a need for the cell division to occur? Now they say that actually, huh, they say that okay, now we will see the significance of mitosis. Now, mitosis actually is responsible for the development of the zygote into an adult. So, when once that mitosis phenomena is over, that process is over. So, what it will do? It will develop into a zygote. And when once a zygote is formed, the zygote under the further will undergo various changes, and it will result in the formation of an adult. So, that is why mitosis plays a very significant role because it will turn into a full-fledged adult. The next equal distribution of the chromosomes actually will be inherited or it will be passed on to each daughter cells. So the distribution of the chromosomes actually will be equal. Suppose two daughter cells are formed means that means equal distribution of the chromosomes will take place. Then it is responsible for the growth and development of an individual. So it is very essential, just I told you, because it will lead to development of zygote. Zygote will develop into an adult. So that means it is responsible for the growth as well as the development of a full-fledged individual. It maintains constant number of the chromosome in all the body cells of an organism. And mitosis is required for asexual reproduction. So always it is required for asexual reproduction, vegetative propagation in the plant, and is also responsible for the repair as well as the regeneration of the damaged tissues. So always, if you observe mitosis, because meiosis actually is a reduction division where the chromosome number is reduced to half, but here it is an equational division where the chromosomes actually are distributed equally. So that is the reason why the mitosis actually plays a very, very important role, and it is required basically for the asexual reproduction, vegetative propagation in the plants, as well as it is also responsible for the repair as well as the regeneration of the damaged tissues. Then mitosis also helps in maintaining the purity of the genome. It also helps in maintaining the purity of the genome as no recombination or crossing over will take place. So in this mitosis, actually it helps in the purity of genome because there is no recombination as well as there is no crossing over. Because due to crossing over and due to recombination, what happens is the genetic material will be exchanged from the parent to the offspring. But since here in case of the mitosis, what happens is it will lead to the formation of the purity of gametes, means it will give rise to pure gametes because of the lack of recombination or the crossing over. Because crossing over or the recombination is a process which occurs only during the meiosis or the reduction division, where actual what is transfer of the genetic material from the parent to the offspring will take place. But here, the recombination of the crossing over does not take place. So that is why meiosis helps in maintaining the purity of the genome because of the lack of recombination. And it is also responsible for the repair and regeneration of old as well as the damaged cells. So it also helps in the repair as well as the regeneration of the old and damaged cells in the animals, like gut epithelium, blood cells, and all those things. So that is why mitosis plays a very, very important role. Now, this is all about the mitosis. Now, we will see what is the meiosis. Now, coming to meiosis. Now, as you all know, actually, basically, there are two types of division I told you, that is mitosis as well as meiosis. Of course, there is also mitosis, where the amitosis, which is also called as amitosis. Now, amitosis actually is not a, cell division which occurs in almost all the cells. Only in the primitive invertebrates you can see, or the single cell organisms, the mitosis will take place, and it is not uniform. So that is the reason why it is not considered to be a what do you say a specific. I mean. Type of cell division. But whereas mitosis and the meiosis actually are the regular, you can say, phenomena which occurs in the plant cells as well as in the animal cells, and they play a very, very important role in the both in the plants as well as in the animals. Now, coming to the meiosis, you all know meiosis, just I have already told you, it occurs in the germ cells or it occurs in the sex cells. Whereas mitosis occurs only in the body cells. It occurs only in the somatic cells, except the sex cells. But meiosis occurs in the sex cells, that is the male as well as the female gamete. So, sexual reproduction in organisms takes place due to the fusion of the male and the female gametes. So, the sperm and the egg respectively. So, here meiosis occurs only in the sex cells, we can see. Now, sex cells means they are nothing but they are called as the gametes, where you have got the male gametes as well as the female gametes. So, that is the egg as well as the sperm. You all know the gametes are haploid in nature. They contain only half the number of the chromosomes. They have got only half the number of the chromosomes. So their genetic content makes them different from the other body cells. Okay. So from the other body cells, the other body cells, they contain the full chromosome. But here in this case, the genetic makeup is different from the other body cells because they contain only half the number of the chromosomes, and they are haploid in nature. They are haploid in nature. So meiosis actually leads to the formation of nothing but the haploid cells. So we can say that meiosis leads to the formation of the haploid cells. Now we will see how this meiosis will take place. Now, mitotic cell division actually is equational in nature, while meiosis actually is a reduction division. So the salient features of meiotic division that make it different from mitosis are as follows. Now we will see how this meiosis will differ from that of the mitosis. One major difference is meiosis occurs in the sex cells, and it leads to the haploid number, or it gives rise to the haploid number of the chromosomes. That is one thing. Now see one by one what are those. So they say that actually it occurs in two stages of the nuclear and cellular division. That is what we call it as meiosis 1 and meiosis 2. So it occurs in two, or we can say meiosis is of two types. One is called as meiosis 1 and the other one is called as a meiosis 2. DNA replication occurs, however, only once. So here only once the DNA replication will take place. Second thing is it involves the pairing of homologous chromosomes and recombination between them. So it involves pairing. Whereas in just we have seen that in mitosis, there is no pairing or there is no recombination. But here in meiosis, you observe the recombination or the pairing of the homologous chromosomes. And here 4 haploid daughter cells are formed at the end, unlike 2 diploid daughter cells that is found in mitosis. So that is a difference. So here we see 4 haploid cells. We see 4 haploid daughter cells, but in mitosis you see only 2 diploid cells are present, whereas here you see 4 haploid cells. So we can say mitosis actually will lead to the formation of diploid cells, that is 2 in number here, because equal distribution of the chromosomes are there. But in case of the meiosis, it is not equation. Distribution is not equal. Reduction division will take place, and it leads to haploid number of the chromosomes, especially, it forms about 4 haploid cells are being formed, because here the recombination or the crossing over phenomena will take place. Now we'll see one by one what are the different. Now coming to meiosis 1. Now meiosis 1 separates the pair of homologous chromosomes and reduces the diploid cell to haploid. It is divided into several stages that include prophase, metaphase, anaphase, and telophase. Okay. So meiosis 1 will separate the pair of homologous chromosomes. Now we'll see what are the different stages of the meiosis 1. So the different stages of meiosis 1 are occur. So the meiosis 1 actually again is divided into prophase 1, metaphase 1, anaphase 1, and telophase. Already we have seen in case of mitosis, that is prophase, metaphase, anaphase, and telophase. But here, why we are calling it as 1, 2, 1 and 2 is meiosis is of two types. One is called meiosis 1 and the other one we call it as meiosis 2. So in meiosis 1, again, same way like mitosis, there are 4 different stages, like that is prophase, metaphase, anaphase, and telophase. Whereas in case of meiosis also, you have two different what say the meiosis 2 also you have got prophase, metaphase, anaphase, and telophase. So to actually differentiate between them, they have named it as prophase 1, metaphase 1, anaphase 1, and telophase 1 for meiosis 1, and for meiosis 2 they have called it as prophase 2, metaphase 2, anaphase 2, and telophase 2. Now we'll see one by one how actually it occurs. Now coming to meiosis 1 stages. Now the different stages of meiosis 1 are prophase 1, metaphase 1, anaphase 1, and telophase. Now we will see one by one what actually happens. Coming to prophase 1, the prophase 1 is longer than the mitotic prophase. It is longer than the mitotic prophase, and it is further divided into 5 substages. Whereas in case of the mitosis, we have not seen this. We have just said prophase, but prophase 1 actually is longer, and it is again further subdivided into 5 substages. Like you have leptotene, zygotene, pactine, diplotene, and diakinesis. What are the leptotene, zygotene, pactine, diplotene, and diakinesis? Now let's see what happens. So here the chromosomes will begin to condense and attain a compact structure during the leptotene. So in the leptotene, what happens? As we have seen in the prophase, so here also the chromosomes will try to condense. They will try to come close together. They condense and they try to attain a compact structure that actually happens in the leptotene stage. Then in zygotene, what happens? In the zygote, the pairing of the homologous chromosomes and the chromosomes will start a process known as the chromosomal synapses. So here, actually, in the zygotene, the pairing of the homologous chromosomes will take place, which is called as nothing but the synapse, accompanied by the formation of a complex structure called as synaptonymal complex. So here, the pairing will take place in the zygotene, occurring in the formation of the chromosomal synapse, which is again accompanied by the formation of a complex which we call as synaptonymal complex. Apart from this, a pair of synapsid homologous chromosomes will form a complex known as a bivalent or tetrad. So apart from this, you also see that a pair of synapsid homologous chromosomes, what do? Is they try to occur either bivalent, that is in 2 forms, or either in a tetravalent, that is either tetrad or a bivalent form. These are the things that we see in the zygotene. Now coming to the next stage, that is a pachytene. In pachytene, the crossing over of the non-sister chromatids or the homologous chromosomes will occur at the recombination site. So in this pachytene, what happens? The crossing over phenomena of the non-sister chromatid of the 2 homologous chromosomes will take place, and these chromosomes will remain linked at the site of the crossing over. So they try to link at the site of the crossing over. Next is what? Diplotene. Now, diplotene marks the dissolution of the synaptonymal complex. So during this particular period, the synaptonymal complex which has been formed, it totally will get dissolved, okay, and the separation of the homologous chromosomes of the bivalent except at the sites of the crossing over. So not only the synaptonymal complex will get dissolved, but at the same time, the homologous chromosomes also of the bivalent, they also get separated except at the point of crossing over, because at the point of crossing over, they try to exchange the nuclear material. So only at that particular the site only they will not get separated, but the other part they will get separated. So the X-shaped structures are formed or are seen during the separation, which are called as nothing but the chiasmata. So the X-shaped structure that you observe during this particular phase, you call it as nothing but the chiasmata. And the last stage you call it as nothing but the diakinesis. Now, diakinesis actually is marked by the termination of chiasmata and the assembly of meiotic spindle to separate the homologous chromosomes. The nucleolus again will disappear and the nuclear envelope will break down. So, in the diakinesis, what happens? The chiasmata will get terminated. The chiasmata will get terminated, and the assembly of the meiotic spindle to separate the homologous chromosomes. So, the meiotic spindle will assemble to separate the homologous chromosomes. Further, the nucleolus also will disappear, and also the nuclear envelope will break down. So as a result, what happens, as we have seen in the prophase, the chromosomes will get distributed in the cytoplasm. So all these occurs in the nothing but in the prophase 1. Now coming to metaphase 1. In the metaphase 1, the bivalent, they align at the equatorial plate, and the microtubules will form the opposite poles attached to the pairs of the homologous chromosomes. As we have seen, same way, here also the bivalent pairs which are there, they will be present at the equatorial plane, and the microtubules from the opposite poles, they get attached to the pairs of the homologous chromosomes. Then in the anaphase, the 2 chromosomes of each bivalent will separate and they move towards the opposite poles. So the 2 chromosomes of each bivalent, they will get separated and they'll move to the opposite poles of the cells. And the sister chromatids actually are attached to each other. Then in the last one, that is what we call it as a telophase 1. In the telophase 1, the nuclear membrane again will reappear and it is being followed by cytoplasm. kinase, and this will give rise to a dyad of cells. So that means it will give rise to two cells. So that is how we have that is about the meiosis one. So meiosis, since it is of two types, we are calling it as meiosis one and meiosis two. Now, meiosis, as you all know, it occurs in the sex cells or in the gametes, that is the sperms as well as the ova. It is divided into four phases, like you have prophase one, metaphase one, anaphase one, and telophase one. Prophase one, we have seen it is the longest phase. Again, it is divided into leptotin, zygotin, paclitatin, diplotin, and diakinases. And we have seen what are the different stages, what is it, the changes that occur in these, followed by metaphase one, anaphase one, and telophase one. So that is how. So at the end of the telophase one, you actually this meiosis one will give rise to a dyad of the cells. Now, coming to the significance. Now, what is the significance of this meiosis one? They say that actually it reduces the chromosome number to half of the sexual reproduction. It will reduce. Whereas in mitosis, we have seen it is an equational distribution. Equally, the chromosomes are distributed. But here, since it is called as a reductional division, it reduces the chromosome number to half. And the crossing over will lead to variations. Whereas in mitosis, you don't see crossing over. But here you see the crossing over. So it will lead to variation. And then random distribution of paternal and maternal chromosomes will also lead to variation, because here random distribution will take place. It is not uniform. So random distribution of the parental as well as maternal chromosomes will lead to variation. And here there will be a disturbance in the disjunction causes genomatic mutations. So since there is a disturbance in this, what happens? It will cause a mutations. And the formation of the spores or the gametes is also induced. So because of that, the formation of the spores or the gametes will also get induced. So this is about the significance of the meiosis one. Next, we will see about the meiosis two. Now, as you all know, just now I have already told you that meiosis is of two types. One is called as meiosis one, and the other one is called as meiosis two. Now, here they say that meiosis two is just similar to that of mitotic division. It is just similar to that of the mitotic division, but not mitosis, because it always occurs in the haploid cells. It is not preceded by interphase, and the two chromatids of the chromosomes are often dissimilar, and the daughter cells formed after meiosis 2 are neither similar to each other nor similar to their parent cell. So that is a difference. They say that the divisions, with reference to divisions and all these things, it is okay, but not mitosis, because mitosis actually always occurs in the haploid cells, whereas this one occurs in the diploid, that is the sex cells, and it is not preceded by interphase, whereas mitosis is always followed by the interphase stage. And the two chromatids actually of a chromosome are often dissimilar, and the daughter cells that are formed after meiosis 2, they are neither similar to each other nor they are similar to their parent cell. Now we will see now the various stages of the meiosis 2. Now coming to the various stages, meiosis 2 also comprises of 4 stages and are relatively simple as compared to meiosis 1. And these 4 stages are prophase 2, metaphase 2, anaphase 2, and the telophase 2. What are they? Prophase 2, metaphase 2, anaphase 2, and the telophase 2. Now, now we will see what actually happens in the prophase 2. Now, the prophase 2 actually is immediately, it immediately sets off after cytokinesis when the daughter cells are formed. So, once the meiosis 1 is over, that is cytokinesis of meiosis 1 is over, then the prophase 2 immediately will start. So in this, what happens when the daughter cells are being formed? So immediately it sets off after the cytokinesis. And here again, the chromosomes, they begin to condense, accompanied by the dissolution of the nuclear membrane and the disappearance of the Golgi operators as well as the ER complex. So in this case, what happens again the same way? The chromosomes, they begin to condense, they try to attain the shape, followed by the dissolution of the nuclear membrane as well as the disappearance of the Golgi apparatus as well as the endoplasmic reticulum. So these are the changes that you see in case of the prophase 2. Now the prophase 2 is followed by metaphase 2. Now here again, the chromosomes are connected to the centriole poles. In the case of metaphase, the chromosomes are connected to the centriole poles at the kinetochores of sister chromatids through the microtubules. They also get aligned at the equator to form the metaphase plate. So in the metaphase, actually, what happens? The chromosomes, they come and settle in the equator. So they form a sort of plate, which is what we call it as, I think, metaphase plate. Metaphase plate. As we have seen in metaphase 1, here also the same thing we observe. Now coming to the anaphase 2. Now in the anaphase 2, in this phase of meiosis 2, there is a simultaneous splitting of the centromere of each chromosome. So what happens? The centromere of each chromosome, it will split, and the sister chromatids actually are pulled away towards the opposite poles. So when once the centromere, because what is the role of centromere? To hold the chromatids, or to hold the sister chromatid. So when once that centromere of the chromosome gets splitted, definitely the two sister chromatids will be free. So what happens is the two sister chromatids, they will be pulled towards the opposite poles. They will be pulled towards the opposite poles. So as the chromatids move towards the opposite poles, what happens? The kinetochore is at the leading edge with the chromosomal arms trailing. So it will lead with the chromosomal arms trailing towards it. So that is actually what happens in case of the anaphase 2. And the last one is called as the telophase 2. In the telophase 2, again same way, the chromosomes will dissolve again into an undifferentiated lump, and a nuclear envelope develops around it. That means again here what happens is the chromosomes, they try to become, decondensed. So they try to attain their original shape, whatever it is. So as a result, what happens? The nuclear envelopes will try to form around it, or it try to develop around it. So as a result, what happens? The cells will be formed. So these are the things that occur in the telophase 2. So since we have seen that meiosis 2 is similar to that of the mitosis, but there are certain what do you say, the things that did not resemble with that of the mitosis, because mitosis, we can say, is totally different from that of the meiosis in certain aspects. Like what we have observed, that in mitosis occurs in haploid cells, but meiosis occurs in diploid cells. Then mitosis is followed by interphase, but meiosis is 2 is not followed by interphase. The next is the two chromatids actually are dissimilar, but here we see they are similar, and the daughter cells that are formed after meiosis 2 are neither similar to each other, nor they are similar to their parent. So these are some of the differences. And the stages, of course, are similar to that of the mitosis, as we have seen, like prophase 2, metaphase 2, anaphase 2, and the telophase 2. And actually, what happens in all these, we have already known. In prophase 2, actually occurs soon after the cytokinesis, when the daughter cells are being formed. And what happens? The chromosomes will begin to condense, followed by the dissolution of your nuclear membrane, as well as the disappearance of the Golgi apparatus and endoplasmic reticulum. Then this is followed by metaphase 2. In the metaphase 2, what happens? Since the chromosomes are connected to the central poles at the kinetochore of sister chromatid to microtubule, they also get aligned at the equator. So we can say that actually in case of in the metaphase 2, the equatorial plate is being formed, or which is what we call it as nothing but the metaphasic plate. Then next you have the anaphase. In this anaphase, what happens is there is a simultaneous splitting of the centromere. So the centromere actually will get split. Each chromosome centromere will get split. As a result, what happens? The histochromatids also will be released, or they get separated. And as a result, what happens? The histochromatids, they are pulled away towards the opposite poles. And as these chromatids they move towards the poles, then the centromere or the kinetochore, which is at the leading edge with the chromosomal arms, also get trailed. And last is your telophase stage, where again the chromosomes will dissolve, means they will again decondense, they will dissolve again, and they develop into an undifferentiated lump. And as a result, what happens? The nuclear envelope will develop around it to form a new cell. So that is what happens in the telophase 2. So this telophase again, similarly again, it is followed by cytokinesis that marks the end of the meiosis. So 4 haploid daughter cells are being formed as a result. So in case of first meiosis, only 2 haploid cells are formed, but after the end of meiosis 2, you have 4 haploid cells. So remember that actually the difference between these two, always you should be very, very clear. Though it is almost similar, but we can say that in mitosis occurs in the body cells or the somatic cells, meiosis occurs in the what do you say, sex cells, that is gamete cells, both the ova as well as the sperm. And here it occurs in mitosis occurs in haploid cells, whereas meiosis occurs in the diploid cells. And here equational distribution equally, the chromosomes are being distributed in case of mitosis, but in case of meiosis, they are not equally distributed. There is a reduction division, and it gives rise to four haploid cells. But here you have only two diploid cells are being formed, and these are the differences. But remember that meiosis actually occurs in two stages, that is meiosis 1 and meiosis 2. Meiosis 1 is a is the phase which is divided again into prophase 1, metaphase 1, anaphase 1, and telophase 1. Among them, prophase 1 is the longest, which is further divided into five sub stages. We call it as the prophase, metaphase, leptotin, zygotin, pachytin, diplotin, and diakinesis. And among them, the prophase 1 is the longest, we can say. This is followed by all metaphase 1, anaphase 1, and telophase 1. So at the end of the metaphase, you see only two cells, two diploid cells. Then this is immediately followed by your metaphase meiosis 2, which is again differentiated into prophase 2, metaphase 2, anaphase 2, and telophase 2. So it is similar to mitosis, but it differs in certain aspects. That's what we have seen. And telophase again is being followed by cytokinesis that marks the end of the meiosis. So as a result of meiosis 2, four daughter cells, that is the four haploid daughter cells are being formed as a result. So that is what we have to see here. This is meiosis 1 and this is the meiosis 2. See here in meiosis 1, this is the interphase 1. You have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1, where you get two cells. Now coming to here. So different stages of meiosis 1 and meiosis 2. Now this is meiosis 2 again. Now here again you have prophase 2, metaphase 2, anaphase 2, and telophase 2, followed by cytokinesis. So these are the difference. So significance of meiosis is it is responsible for the formation of sex cells, okay, or the gametes that are responsible for sexual reproduction. And it activates the genetic information for the development of the sex cells and also deactivates the sporophytic information. It maintains constant number of the chromosomes by halving or by reducing the same. So this is important because the chromosomal number doubles after fertilization. So this is the significance of meiosis. Okay. Now we'll see. The next is what? Here in this process, independent assortment of maternal as well as the paternal chromosomes also take place. Thus, the chromosomes and the traits are controlled by them are reshuffled. And the genetic mutation occurs due to the irregularities in the cell division by meiosis, whereas in mitosis, you don't find it. And the mutations that are beneficial are carried on by the natural selection. And the crossing over will produces a new combination of traits and variations. Whereas in case of mitosis, you don't find, but only because of the crossing over phenomena, you actually have got the variations. Now we will see now. So with this, we have completed what? Mitosis as well as meiosis. Now we will see what are the differences that occurs between the mitosis and meiosis. Already we have seen, but still once again we will see. Now coming to mitosis, now first interphase, prophase, metaphase, and telophase. So in the interphase, each chromosome will replicate during the phase of interphase. The result is two genetically identified sister chromatids actually will be formed, but they are not a part of mitosis because it takes place between one mitotic phase and the next. Now here in case of meiosis, the chromosomes not yet visible, but DNA has been duplicated or replicated. Then in the case of prophase, each of the duplicated chromosomes appears as two identical or equal sister chromatids, and the mitotic spindle will begin to form chromosomal split condense and thicken. Whereas here in meiosis, prophase 1 crossing over and recombination, homologous chromosomes appear together as phase. Tetrad or bivalent is a structure that is being formed. Segments of the chromosomes are exchanged between non-sister chromatids at crossing over point known as chiasmata. Whereas in case of metaphase, the chromosomes assemble at the equator of the metaphase stage. Here also in metaphase 1, chromosomes adjust on the metaphase plate. Chromosomes are still intact and arranged as pairs of homologous bivalent. Then in the anaphase, the spindle fibers begin to contract. This will start to pull the sister chromatid apart. At the end of anaphase, a complete set of daughter chromosomes is formed. Whereas here, in case of meiosis, sister chromatids may stay intact. However, homologous chromosomes drift to the opposite poles to reverse the course. And nothing. The next is what? Mode of reproduction. If you observe, In case of mitosis, it is asexual reproduction. In meiosis, it is sexual reproduction. And then occurrences, it occurs in all the somatic cells, whereas meiosis occurs only in the reproductive cells or the sex cells. And function of the mitosis is general growth and repair and cell reproduction, whereas in case of meiosis, it is genetic diversity through sexual reproduction. And cytokinesis occurs in telophase in case of mitosis. Here also, it occurs in telophase 1 and telophase 2. And mitosis actually was discovered by Fleming, Walter Fleming, whereas meiosis actually is discovered by Oscar Hertwig. So this is all about the cell division and the cell cycle that we have seen. So both mitosis as well as the meiosis are very, very important. So with this today, now we will end the class. Tomorrow again, we will meet and start with the new chapter.